Introduction. Scenes: a lecture hall filled with medical students. Professor Smith stands at the front of the room, preparing to deliver a lecture on Huntington's disease. Professor Smith: Good morning, everyone. Today, we will be delving into the fascinating topic of Huntington's disease, a devastating neurodegenerative disorder that affects thousands of people worldwide. This comprehensive lecture aims to provide you with an in-depth understanding of the various aspects of Huntington's disease, from its underlying genetic causes to potential therapeutic strategies. So, without further ado, let's begin. Scene: a slide is projected on the screen, displaying the structure of DNA. Professor Smith: Huntington's disease is primarily caused by an abnormal expansion of trinucleotide repeats in the Huntington gene, located on chromosome 4. These repeats consist of the nucleotides cytosine, adenine, and guanine, abbreviated as CAG. Normally, this region contains 10 to 35 CAG repeats. However, in individuals with Huntington's disease, the CAG repeats exceed 36, leading to the production of a mutant Huntington protein. Scene: a diagram illustrates the process of trinucleotide repeat expansion. Professor Smith: The mechanism underlying this trinucleotide repeat expansion involves genomic instability, specifically DNA slippage during replication. This instability results in an increased number of CAG repeats with each successive generation, leading to an earlier onset and more severe symptoms in affected individuals. Scene: a slide shows an illustration of protein aggregates known as inclusion bodies. Professor Smith: The mutant Huntington protein tends to misfold and aggregate in neuronal cells, forming inclusion bodies. These inclusion bodies are composed of insoluble and toxic protein aggregates, which interfere with vital cellular processes. Scene: a microscopy image reveals the presence of inclusion bodies in brain tissue. Professor Smith: The accumulation of these inclusion bodies disrupts essential cellular functions, such as protein degradation, mitochondrial dynamics, and synaptic transport. Consequently, neurons in the striatum and cortex, particularly medium spiny neurons, become progressively damaged, leading to the characteristic motor and cognitive symptoms of Huntington's disease. Scene: a slide displays a mitochondrion, highlighting its various components. Professor Smith: Research has shown that mitochondrial dysfunction plays a crucial role in the pathogenesis of Huntington's disease. Mitochondria, often referred to as the powerhouses of the cell, are responsible for generating adenosine triphosphate, ATP, through oxidative phosphorylation. Scene: a graph illustrates the decrease in ATP production in Huntington's disease. Professor Smith: In individuals with Huntington's disease, neurotoxicity caused by the mutant Huntington protein disrupts mitochondrial function, leading to impaired ATP production and increased oxidative stress. This mitochondrial dysfunction further aggravates neuronal damage, exacerbating disease progression. Scene: a slide shows an image of activated microglia, the immune cells of the brain. Professor Smith: In addition to the genetic and cellular factors, neuroinflammation also contributes significantly to the pathogenesis of Huntington's disease. Activation of microglia, the resident immune cells in the brain, triggers an inflammatory response. Scene: a diagram depicts the release of pro-inflammatory cytokines. Professor Smith: This response involves the secretion of pro-inflammatory cytokines, such as tumor necrosis factor alpha, TNF-alpha, and interleukin-1 beta, IL-1 beta, which further perpetuate neuronal damage by affecting synaptic plasticity, promoting excitotoxicity, and activating additional inflammatory cascades. Scene: a slide displays various imaging techniques, such as MRI and PET scans. Professor Smith: Identifying reliable biomarkers for Huntington's disease is crucial for accurate diagnosis, monitoring disease progression, and assessing the efficacy of potential treatments. Both structural and functional imaging techniques offer valuable insights into the extent and location of neuronal damage. Scene: a graph demonstrates the correlation between a specific biomarker and disease severity. Professor Smith: For instance, magnetic resonance imaging, MRI, can reveal structural changes in the brain, including the loss of brain volume and progressive cortical thinning. Positron emission tomography, PET, scans can measure glucose metabolism and neuroreceptor activity, providing functional markers that correlate with disease severity. Scene: a slide shows the gene suppression techniques. Professor Smith: Gene suppression strategies aim to reduce the production of the mutant Huntington protein by targeting the mutant allele specifically. Scene: a diagram illustrates the use of RNA interference, RNAi, to target the Huntington gene. Professor Smith: One approach involves utilizing RNA interference, RNAi, to degrade the mutant Huntington mRNA and prevent its translation into the toxic protein. Another approach involves utilizing antisense oligonucleotides, ASOs, to selectively degrade the mutant Huntington mRNA or inhibit its translation. Scene: a slide presents a chart depicting the varying degrees of cognitive impairment in Huntington's disease. Professor Smith: cognitive dysfunction is a prominent feature of Huntington's disease, although its severity and specific manifestations can vary widely among individuals. This heterogeneity poses challenges for clinicians and researchers in accurately assessing and managing cognitive impairments. Scene: a diagram illustrates the associated cognitive domains affected in Huntington's disease. Professor Smith: the cognitive impairments observed in Huntington's disease encompass multiple domains, including attention, executive function, working memory, visuospatial abilities, and language. Understanding the underlying mechanisms of these cognitive deficits can potentially guide the development of targeted therapeutic interventions. Scene: a slide displays a diagram representing the HD lipidome. Professor Smith: HD lipidome refers to the lipid profile of high-density lipoprotein, HDL, commonly known as good cholesterol. Recent studies have shed light on the role of HDL and its lipid components in modulating the pathogenesis of Huntington's disease. Scene: a graph depicts the dysregulation of specific lipid species in Huntington's disease. Professor Smith: dysregulation of various lipid species, including phospholipids and sphingolipids, has been observed in individuals with Huntington's disease. Alterations in HDL composition and function have implications for neuronal health and survival, influencing disease progression and potentially providing novel therapeutic targets. Scene: a slide displays the CRISPR-Cas9 gene editing system. Professor Smith: advances in gene editing technologies, such as CRISPR-Cas9, hold promise for the treatment of Huntington's disease by directly targeting and modifying the mutant Huntington gene. Scene: a diagram illustrates the CRISPR-Cas9 system editing the Huntington gene. Professor Smith: CRISPR-Cas9 enables precise genomic modifications, either by excising the expanded CAG repeats or by introducing corrective DNA sequences. Despite the complexity and challenges involved in implementing this approach, gene editing strategies represent a potential avenue for long-term therapeutic interventions. Scene: a slide shows a flowchart representing the sequential events in the neurodegenerative cascade of Huntington's disease. Professor Smith: the pathophysiology of Huntington's disease involves a complex cascade of events that ultimately leads to widespread neurodegeneration. Scene: a diagram describes the sequential involvement of various cellular processes. Professor Smith: dysregulation of key cellular processes, such as transcriptional regulation, protein quality control, neurotransmission, and axonal transport, initiates a chain reaction of cellular dysfunction, culminating in neuronal death. Understanding and targeting these specific steps in the neurodegenerative cascade are crucial for developing effective therapeutic interventions. Scene: a slide displays the comorbidity between Huntington's disease and psychiatric disorders. Professor Smith: Huntington's disease often presents with psychiatric symptoms that significantly impact the quality of life for affected individuals and their families. Scene: a graph illustrates the prevalence of psychiatric symptoms in Huntington's disease. Professor Smith: psychiatric manifestations, including depression, anxiety, irritability, apathy, and psychosis, add an additional layer of complexity to the clinical management of Huntington's disease. Understanding the intricate interplay between the neurodegenerative processes and psychiatric symptoms is crucial for developing comprehensive care plans. Scene: a slide displays the concept of precision medicine. Professor Smith: precision medicine aims to tailor treatment strategies based on an individual's unique genetic and molecular profile. Scene: a diagram illustrates the integration of genetic information, biomarkers, and therapeutic approaches. Professor Smith: in Huntington's disease, precision medicine holds tremendous potential for customized therapeutic interventions, enabling targeted therapies that address specific pathophysiological mechanisms or genetic modifiers. Professor Smith: in conclusion, Huntington's disease is a multifaceted disorder influenced by genomic instability, protein aggregation, mitochondrial dysfunction, and neuroinflammation. Identifying biomarkers, developing gene suppression strategies, and understanding the cognitive heterogeneity are crucial for effective management. Advancements in gene editing, precision medicine, and our comprehension of the neurodegenerative cascade further augment our efforts to combat this devastating disease. I hope this lecture has provided you with a comprehensive understanding of Huntington's disease and its myriad aspects. Scene: the students applaud as Professor Smith concludes the lecture.